स्टार्ट कर दूं?
हम आप ही करिए।
ठीक है।
We are live.
So good evening children, I hope you can hear me. A very good evening to all of you, and I hope you're in the best of the spirits and preparing well for your NEET exams. Now it's very important to keep your spirits up. Well, here in North India it's really cold and it's difficult to actually keep going with high spirits and keep warm. That's what I need to tell you. So from wherever you are, from Patna, from Mumbai, from whichever part of the country you are, you have all my best wishes, and let's get on together to analyze the question paper we are here to analyze today. Now I hope you have all done well. I will start with the congratulating with the toppers of today. Ruchir Gupta with 100 marks. I would definitely say this is a very good score, Ruchir. Just keep it up. Praloy Saha is at 96. Really very good. Again, well, 4 marks may be a matter of chance. And yet, Pranoy, sometimes it may not really be a matter of chance, but a matter of being really careful about one of the questions. But as you can see, one question can cost you a real big, right? Now, the next is Harsh 95. And definitely again a very good score, Harsh, and I hope you will keep going strong. But yes, do take care to not miss out on the very minute details that can cost you the 5 marks that they have, right? Now Rashi at 91 again. Rashi, my congratulations to you, and keep going really strong. Also congratulations to all the high achievers of today, and a piece of real good advice, and all my I wouldn't say sympathy, but I would definitely say that all the rest of the students need to really pull up the socks and keep working hard. I'm sure the big scores are not very far. And when you analyze this paper with me today, I hope you will not just see on the surface of the questions, on the matter of the questions, by which I mean the biology of the questions, but also tackle the psychology of the questions with me. Now what do I mean by that? Now there are several types of questions, children. I will definitely talk about them as I go on. But there are some questions which I call the mousetrap questions, which are questions with a little bit of a catch, which look very simple and very straightforward, but do have a small catch somewhere that actually pulls you in. Now, as I was analyzing today's your response to today's paper, what I saw was that the mousetrap question got most of the mice. Well, sorry for calling you mice here, but it really goes well with the analogy. So what I mean to say is, don't be mice to be caught in mousetrap questions, which means learn to see the tricks of the questions, right? Now, I will definitely tell you about which question really tricked you most, right? And mind you, this is a question in which I altered one of the choices to create a mousetrap, just knowing that probably it might trap some of you. But what I'm seeing is that it has actually trapped many of you, if not most of you. Well, let us then start on with the questions analysis, and I hope you will keep giving me the response of where I'm falling short of your expectations. If there is something that you need me to elaborate more on, I will definitely do that. It will be my pleasure to explain things to you. And also, if there is something that you need to ask, out of the blue, which means out of the box, then you are most welcome to do that, so long as we keep sticking to the time schedule. Now, I will start with the question that most of you have faltered in, which means the question that 38.89% of you have marked as incorrect, and amazingly, it was one of the easy questions. And however, as I told you, this was the mousetrap question that I created for you. So this question is the 7th question, as I can see here. In the 7th question, it says, in genetic engineering, the antibiotics are used as selectable markers. Can we have the question here on the board? Yeah. Okay, so you can see the question, and what you can see here on the question is that we have the options as selectable markers, that's the first option. The second one says to select the transformants. The third one says as sequences from where replication starts. And the fourth option says to keep the cultures free of infection. Now, I am guessing that you couldn't have confused between the third and the fourth options. It was actually the first and the second options which were the closest options and where the possibility of confusion arose. Now, I'm sure that most of you have probably marked the first one. Am I right? Can you give me a response? Which particular option have most of you marked? My guess is that most of you marked option 1 because this happens to be a phrase directly from the NCERT. So what I was thinking was that it probably, without thinking twice about it, you would probably go off to mark this first option. Yes, so Arjun is telling me that most of you have marked as selectable markers. But children, try to understand the trap here. The selectable marker actually is the gene which is used to help select the transformants and the recombinant transformants. So when we talk about the vector, recall that there is a vector called PBR322 and the PBR322 has two selectable markers. Let me draw the rough diagram line sketch of the PBR322 and you can understand, I'm supposed to draw here, I'm sorry. So this is the PBR322, you know that there is an ampicillin resistance gene here and a tetracycline resistance gene somewhere here. So what is this? You know this to be and this to be a selectable marker. Now what is this? Is this an antibiotic or is this an antibiotic encoding gene? Well, the part of the vector is the antibiotic encoding gene. Let me write it down here for you. Antibiotic encoding gene. So these two are the genes which encode for the antibiotics tetracycline and ampicillin respectively. So these are the selectable marker genes used on the vectors. And when we are talking about using the antibiotics, then we cannot say that these are the markers. Antibiotics are not the markers. The antibiotics are used to select out the transformants from the non-transformants. So here the second choice is more appropriate if you look at the language carefully. So do you all agree with me? Come please. I'm waiting for your response. Do you all agree?
Ashutosh is asking,
yes?
What is the difference between 1 and 2 options?
Yes, that is what I'm telling you, Ashutosh. Now again, let us explain this once more to make it clear. Option 1 says as selectable markers. So when we say selectable markers, this actually refers to the genes which are present on the vector DNA which help us to select out the transformants from the non-transformants. So this means that they are genes, which means that they are segments of DNA. A gene is a segment of DNA, right? So this is a gene which encodes for antibiotic resistance. Now you know that markers can be of two major types. One is the antibiotic encoding genes, as the ones we have just discussed, and the other ones are the visual markers like the LAC-Z or the Betagalactosidase gene, about which you have also studied. It is also there in the NCERT, Ashutosh, just to recall that, right? So these two are actually the genes used for this purpose, and therefore the genes used for this purpose are termed as the selectable markers, right? But when we're talking about the antibiotic itself, which means this is the gene and it is coding for the antibiotic ampicillin resistance. It's not even actually coding for the ampicillin antibiotic. It is coding for some proteins which gives resistance to the ampicillin antibiotic, right? So which means when we put ampicillin on these cells, then the cells will be able to survive if they have this gene, right? So the gene is the marker, but the antibiotic is not the marker. It's actually a substance that you're using to facilitate the marker genes. It is It is with the help of these antibiotics that you're able to select out, but these are not called the marker genes, right? I hope that's clear to you. Is there any other problem?
Ma'am, Anirudh is asking,
Yes, Anirudh.
please explain the difference between transformants and non-transformants.
Alright, transformants and non-transformants. Well, I'll just rub out the board and explain this to you nicely. Now, Anirudh, if you recall from the other sections of your biology syllabus that you have studied, you have talked about transformation that was studied by Griffith. Recall that? When Griffith conducted certain experiments, then he actually converted the live R strains of the bacterium Diplococcus into S strains. Why? Because they managed to pick up the capsule forming gene from the outside, right? Do you recall that experiment? It's there in the NCRT for in the chapters of genetics also. In fact, you must have studied in your diversity chapters in class 11th also. So recall that transformation is a process in which a particular cell can pick up either pick up the DNA from outside voluntarily, which means by its own will, right? Or the DNA can be forced inside a cell, as we sometimes do with the help of gene guns or with the help of vectors or whatever the method be. When any piece of foreign DNA is picked up, taken in and expressed by a cell, then it is said to have transformed. Which means if a particular cell picks up a gene from outside and starts to express it, we say that it is a transformed cell. It is a genetically transformed cell, right? So when we are talking about transformants and non-transformants, suppose these are the two sets of cells that we are talking about. Now in one place we have, we have a recombinant DNA which has a vector and a piece of DNA, or we can actually term it as the desired gene. You know that recombinant DNAs are made by combining a piece of desired DNA with the vector, right? So when we have this recombinant DNA, and when we incubate this vector DNA, recombinant DNA rather, with the cells, some cells will pick this up, right? DNA from outside, and the other cells will not pick it up because it is not mandatory, right? It is not 100% sure that a cell would pick up the DNA from outside or will not pick up the DNA from outside. So if it has picked up the DNA from outside and has started expressing it, we will call this as a transformant, right? And we will call this as a non-transformant, right? So this is the difference between transformant and non-transformant. Once a cell which is able to pick up the DNA from outside, the foreign DNA from outside, is a transformant and of course express it. And if it is not able to pick up the foreign DNA from outside, then it is a non-transformant. Now that's the difference. Now, however, let me tell you here, there is also a difference between a recombinant transformant and a non-recombinant transformant. What I'll do is I'll take 2 minutes more here to actually differentiate between these two also, because it becomes a problem when you don't differentiate between the recombinant and the non-recombinant transformants. So let me pick it on from here. If I have the difference between a transformant and a non-transformant, let me take this up and tell you that even the transformants can be of two types. We can have recombinant transformants and we can have non-recombinant transformants. Non-recombinant transformants. Now, let me tell you the difference between the two. Now, when we incubate the host cells with certain pieces of DNA, then there are different things that might happen, and let's see what are the different things. So I will start on from the initial part only. Now suppose we have a vector here, right? This is a vector and this is my desired DNA, right? I have used the different color codes for that. Now what I do is I cleave this by using a restriction enzyme, right? And by using the same restriction enzyme, I have cleaved this part of the desired DNA also. Now when I put them together, they are likely to join because of their similar sticky ends, right? So if these two happen to combine and form a recombinant DNA, then I have with me a recombinant DNA, and this is the simple vector, right? Now I can make this, right, to go inside the cells, the host cells that I want them to go into, right? For example, these might be the Escherichia coli cells or yeast cells or whatever be the host cell that I am talking about, right? Now suppose I take these cells and this recombinant DNA in a test tube or whatever solution and put them on together and hope that this will be picked up, right? Now there is also a very big chance that instead of these two combining, this snaps back together, which means this combines with itself together. Although we do have certain enzymes that ensure that the two ends of the vector will not snap back together, but there are some possibilities that even this may happen. Now there might be three types of cells which may originate from this incubation, right? So as I told you, one type of cell is a non-transformant which hasn't picked up anything, right? Not picked up anything, right? So it fails to pick up either this or whatever, right? So this is actually a non-transformant. So what the cells which have not picked up anything are non-transformants, right? Now there might be some cells which have picked up the recombinant DNA that I had made, right? So these kind of cells are termed as recombinant transformants, right? And the cells which happen to pick up the vector only, right? As I told you, sometimes the vector might snap back together, or I might have some unbroken vectors in the medium also. So the cells which have picked up the vector only but not the recombinant DNA will also be transformants because they have also picked up something from outside, but they are non-recombinant transformants, right? Now if you recall, there is a very nice example in the NCERT which helps you to actually differentiate between these three types of cells using two antibiotic resistance genes, that is ampicillin resistance gene and the tetracycline resistance gene found on the vector PBR322, right? Now I hope this difference is very clear to you. Cells which haven't picked up anything are non-transformants. Those which have picked up the vector only are the non-recombinant transformants, and those cells which have picked up the recombinant DNA, which means the vector with our desired gene ligated to it, then it is a recombinant transformant. And needless to say, we would actually like to select these out of the rest three, right? Rest of the two, right? And that's where the selectable markers come in, the role comes in, right? So should we now move on to the next question, or is there something else? Tell me. Yes. I'm moving on to the next question now. And the next question is the question number 11. The question number 11 has been marked correctly by only 42% of the people. And let us see what the question is. 11th question says, the colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of. Now I'm wondering why most of you have marked it incorrectly. Let me check the question again. Have I read it correctly, and is this really the right answer? Because this is directly from NCERT, children. Let's see. Non-recombinant bacteria containing beta-galactosidase. Well, no, that's not the answer. Insertional inactivation of the alpha-galactosidase in the non-recombinant. Yes, there seems to be some problem with the option here. Let me just check it out. Alpha-galactosidase and recombinant bacteria. Yes, I think there is a reason why you got confused here, children. And let me just explain this to you. Actually, this should be beta-galactosidase, and that's probably why you have marked this incorrectly, because in the second option, The correct option is, well, no, this is correct. This is correct, completely correct. This is right from the NCERT. In the first option, we do have beta-galactosidase, non-recombinant bacteria containing beta-galactosidase. Now, let us understand this quickly, and let's also understand why you marked this incorrectly. I'm a little surprised about this, but let's see. And you can tell me why this problem happened. Now, we have this E. coli cells, we can call them E. coli host cells, and inside them if we have, now suppose we have this vector, right? We have this vector with a certain gene which is called the beta-galactosidase gene, right? So I am marking this as the vector. What is this? This is the vector. Now in this vector, if you see, this particular gene which is called the beta-galactosidase gene is the visual marker gene, right? This will help us to select out the transformants from the non-transformants. Now let's understand how this happens. Now what is the function of the beta-galactosidase gene? Now this encodes for the enzyme beta-galactosidase, right? Right. Now what does this enzyme beta-galactosidase do? This enzyme beta-galactosidase converts a substrate, yes?
Can you explain this side?
Oh yes. The question is coming out on this side?
Yes, ma'am.
Okay, I've been told to explain on this side, so just give me a minute. So I'm redoing this. Right, so once again, let us see, this is the vector and this is the gene. Which gene? This is the beta-galactosidase gene, right? Which I told you is the visual marker gene, right? Now this encodes for the enzyme beta-galactosidase. Now what is the function of this enzyme? This enzyme catalyzes the conversion of a substrate which is called the X-gal. This is the substrate which is colorless, right? A colorless substrate into a blue colored substance, right? So if there is a bacterium which has picked up only this vector, then what will happen? Then that particular bacterial colony will be able to produce this blue colored substance and would look blue in color, right? But suppose I insert the desired gene in the middle of this marker gene, in the beta-galactosidase gene, then what happens? Now suppose this is the same vector that I have cleaved from right in the middle of this gene, right, and inserted my desired DNA in the middle, making a recombinant DNA. So what have I done now? In effect, I have carried out insertional inactivation of the beta-galactosidase gene, also called the LAC-Z gene. What does that mean? That means that this particular gene will not be able to produce this functional enzyme beta-galactosidase, which means that even if we give this substrate to the colony, then it will not be able to produce this blue colored substance, right? So what does that mean? So here the gene in question is the beta-galactosidase gene, right? So that's the first thing that to remember. And the second thing is that if we have a cell, okay, now we are making two types of cells, two types of host cells. This is the one which has picked up only the vector with the functional gene, and this is the host which has picked up the recombinant DNA, right? So which particular colony would you think to be blue colored, children? Which colony out of these two, this one or this one, would be blue colored? Well, Okay, I'm asking a question. Let me see how you respond to it. Which colony would look blue? Will this look blue or will this look blue? Arjun, are there any answers? Which one? Type A. Let me call this type A. Let me call this type B. So which one would be blue colored? A or B? Come on. No answers?
Type A.
Type A. Yes. Completely correct. Because the gene here is intact. It is not inactivated. So the type A ones. Now, these particular type of cells will be termed as non-recombinant transformants because they have not picked up the recombinant DNA. So these are non-recombinant transformants, right? And these types are the recombinant transformants. So the recombinant transformants would look colorless because they have an inactive gene, whereas the non-recombinant transformants would look blue because they have an active gene. So I hope this much is clear to you. This is straight from the NCERT, right? In all the other options, you don't even have beta-galactosidase. So there was no reason for confusion here, right? So the next question that I'm taking up, I hope is all right with you. If you still have a problem, you can come back because we need to, stick to the deadline. So the next one is question number 15. Let us see the next question. Question number 15. Yes, so question number 15 says TI plasmid transfers work with. The TI plasmid transfer works with monocots only, dicots only, dicots mostly, or monocots mostly. Now here the straightforward answer is dicots mostly. Well, maybe some of you have marked dicots only. Well, let me tell you that the recent researchers have shown that in fact some of the researchers have made it possible for the TI plasmid to infect some of the monocots plants also. So it's safer for us to mark dicots mostly, right? Otherwise, generally in nature, this particular bacterium does not infect the monocots. It's a one actually which infects the dicots only, and that was actually a reason for serious trouble because for the simple reason that I really don't know if you know about this, but the top 5 food yielding crops of the world. Now what am I saying here? Which means the top 5 crops which provide food to us, out of the top 5, 4 are monocots. So if we need to transform our agriculture, we need to have some method by which we can transform the monocotyledonous crops also. So now TI plasmid mediated transfers of the gene are very efficient, very effective, but unfortunately they do not work with the monocots because this bacterium does not infect the monocots. But now some recent researchers so scientists have actually somehow wired up the TI plasmid also in such a way that now some of the monocotyledonous plants have also been transformed using the TI plasmid. So here that's the reason why we have marked third as the answer. So I hope you're with me here. And still, if you have a problem, you can come back, right? So the next question happens to be question number 4. So question number 4 is the question which is marked correct by only 50% of you. Now children, keep analyzing, see, this just like the first question was a very mousetrap type question, right? You understand that. There was a small, twist of words, very small twist of words. Also, there is something I need to tell you here, children. Sometimes, you actually need to go into the psychology of the person who's setting up the paper. Now, try to understand, there are a few lakhs of children sitting every year for the NEET exams, right? And the syllabus is the same, right? They can't really, the person who is actually making the question paper cannot really go much beyond the purview of your textbooks, right? So what does that person do? That person will have a psychology to twist that information in such a way to create small traps for you here and there. So if you really haven't been reading between the lines and if you haven't been reading very carefully, you are very, very likely to make the mistakes, right? So just because, sometimes a question may appear very, very simple, okay, this phrase is given in NCERT, so let me just not even think twice about it. Let me mark option 1 because it's given there. Antibiotic, this is a selectable marker, antibiotic, selectable marker. It all obviously means the same thing. It connects. But no, dear, you really have to look closely. There might be a trap there, So if there is a particular phrase in the NCERT, it doesn't necessarily mean that two phrases have to be connected in the same manner as you are thinking about, right? So this is again a very big trap used by the I would really call the paper setters to be big psychologists, They actually play on our psychology, on the student's psychology. What they do is they create traps by using, there's a technique that is used which is called the familiar phrase trap. I call this the familiar phrase trap, which means they would pick out a very familiar phrase from the NCERT, right? And just, use another word from the NCERT, and they know that the student would automatically link the two up, because if you're not thinking deeply, you will definitely do that. But it really doesn't link up, right? So just don't think this is given in NCERT, this phrase is given in NCERT, this word is also given somewhere around that, in the same paragraph this phrase is there, in the same paragraph that word is there, which means this is the right answer. No, please look very carefully, try to understand the language of the questions, right? So now I'm taking up the question number 4. Yes, I am with you on question number 4. Now children, feel free to talk to me through that if you have any problems. In fact, your problem may not be directly to do with the questions, it may be a psychological problem also. Let's discuss that also. Yes, tell me.
Ashutosh is asking, what is hybridoma?
Oh, hybridoma. Ashutosh, what I will do is I will write your doubt somewhere here. But I will take it up after I have finished up the questions first, because it is, although related to biotechnology chapter 2, we will definitely take that up if we have the time. But just to tell you a little bit, these are the fusion cells. These are fusion cells which are formed by the fusion of beta lymphocytes and cancer cells, myeloma cells, and they're used for making monoclonal antibodies. But the details I will not tell you right now, because otherwise we will run out of time, right? But if we are left with time, I will tell you the detail today, otherwise in the next section. So the next question, this question says the cloning vector M13 has the genetic material. Yes. Now you may also ask me, why in the first place did I put this question on the paper? You might also ask me, am I supposed to know the genetic material of all the viruses, all the vectors that are there in the world? Well, let me tell you this. I have selected out the questions from the questions that have appeared in the past in the medical examinations. If you want, I can even tell you the year and which particular medical examinations. So the reason for my putting this here is that this particular question has appeared in the medical examination in the past, and therefore it is worth knowing what M13 is. Although generally the vectors given in your NCERTs and some of the reference books are pretty enough for you to go into NEET, but if there is something which has appeared in the past and we don't know about it, we ought to know about it. So the vector M13 has a genetic material single-stranded DNA. Now what is this M13 vector? First let me tell you, M13 actually is a bacteriophage, right? And if you recall doing the viruses in class 11th very carefully, then you might recall that you studied M13 as one of the examples of the viruses having single-stranded DNA as the genetic material, along with maybe 5X174 virus. The names of these viruses sound like some Sten guns maybe, but then that's how they are named. So this M13 is actually a virus which has single-stranded DNA as its genetic material. Now let me tell you a little bit more about it. We do use M13 for some of the gene transfers, although it's not a very big vector. I will just tell you a couple of things about it since it's been mentioned here. Oh, I rubbed out Ashutosh's hybridomas. Okay, so let me write them back here. So M13, as I told you, is actually a bacteriophage, right? Now this bacteriophage M13 is one which infects only the F plus bacteria. F plus, you recall what are F plus and F minus, children, from your class 11? These are the, the so-called males or the donors in the process of conjugation, right? And actually the infection takes place through the pilus or the sex pilus, and that is the reason why it can affect only the F plus cells, right? Now this vector is relatively a small vector and can actually carry a gene load. I hope you understand what we mean by gene load. Now the gene load means, or the DNA load means, the length of DNA that a vector can carry and deliver into a host cell, right? So this can carry somewhere about up to, I would say about 2 kilo base pairs, right? It can go a little bit more than that also, but then the delivery becomes much less efficient. So up to 2 kilo base pairs is the good load that it can carry. So it's not a very heavy load carrier vector, but it definitely has some of the advantages, like it can actually lead to the cloning of both of the strands, right? Okay, of course we can give a little more explanation here why both of the strands, but I think this is actually beyond the I would say context here, and would lead to more confusions, right? So here the answer becomes 1, that is single-stranded DNA. So since this is a virus and it's a viral vector, and that virus has single-stranded DNA as its genetic material, so the answer here is 1. So you could have connected here. Now this is a question here, I would say, you may say this is not very fair to ask us, but if you recall some of the examples that you've done in class 11th, you could have connected, right? So anyways, now for future, now you know this. Now the next question I'm taking up is 22 and 16. So let me do 16 first. Both of these have the success rate amongst you as about how much? 56, about 56%. Right, so let me see what made you do that wrong. So the 16th question says, the vector used for introduction of the largest genome among the following to the host cells is lambda, M13, cosmate, and YAX. Now I hope you have studied a little bit more in detail about the vectors. Well, there are actually so many vectors that to know the details of all of them is probably not a very practical solution. But what I would suggest to you is that the vectors mentioned in the NCERTs at least are the ones that you must do carefully and read up more about, even beyond NCERT. We know of the vector YAC and BAC mentioned in the Human Genome Project in the 2nd chapter of genetics. Are you able to connect? Yes? So YAC and BAC are the 2 most commonly used vectors in the Human Genome Project. Recall that, right? So although this question is not very directly from this particular chapter, but children, don't ever expect the questions to be straight from a single chapter asking you not to, connect with the other chapters. Now your ability to do your questions well is your ability to link the information given through or across various chapters, across various subjects. Now what do I mean by that here? Now, when you are studying for a particular chapter in biology, when you sit down to answer the questions, right now of course you are not very well equipped to answer across chapters and subjects because you probably haven't finished up the syllabus as yet, or maybe you have. Well, it's actually high time you should have, right? So if you're sitting down to a paper on say chapter 10 of biology, do you really only think about the chapter 10 of biology while answering it? Suppose you have a question in front of you, and when you're answering it, are you going to recall the facts and the figures and the information given only in that chapter? If you're doing that, then I must say that you're doing wrong, because you must be able to link that information across chapters and subjects. You must think that where have I read this? Is it only in this chapter? And if not in this chapter, where else have I read about it? And is it possible that I read about this concept somewhere, maybe in physics also, and somewhere in chemistry also? So if there is a particular concept given in biology and it is connecting with a concept in another chapter in biology or another chapter in chemistry or physics, you must be able to quickly run your mind through and connect all the pieces of information and put them together like a puzzle. If you are able to do that, then I will definitely say you will be a very successful, aspirant for the NEET. So here yak is your vector from the second chapter of genetics, and answer here is yak only. And the reason for this is it's actually one of the most used vectors in the Human Genome Project for its ability to carry big gene loads, for delivery. But of course, let me tell you some of the facts and figures here. The lambda phage is able to carry, the lambda vector is able to carry about 10 to 20 kilo base pairs. So KBP, here means kilo base pairs. Just as you have the gigabytes and the megabytes and kilobytes in your computer language, which I don't know much about. But here in genetics, when we talk about KBP, it actually means kilo base pairs. And you know what a kilo means? Kilo means 1000, right? So a lambda phage can carry a load of 10 to 20 kilo base pairs, and as I told you, M13 can carry about up to 2 kilo base pairs. So it's a relatively small vector, right? And cosmids can carry somewhere about 35 to 45 kilo base pairs. So up to that limit, they can carry the genetic load. What about the yaks? The yaks are the what is the full form of yak? Can anybody tell me quick? I'm waiting. I'm just giving you exactly 5 seconds. Come to me with the answer quickly. Arjun, are there any answers?
There's a latency of like about 13 seconds.
Alright. So we'll have to wait for 13 seconds for the answer?
Yes, ma'am.
Alright, then let me not wait because we'll be wasting a lot of time. So anyways, if the answers come up, do tell me. If somebody is able to give me the right answer for the full form of yak, right? So yak, till then I will refrain from answering you, and this yak actually
yeast artificial chromosome.
Oh yes, yeast artificial chromosome.
Rashi, Rahul.
Great.
Lakshika.
Rashi, Rahul and all the others who have given the right answer, congratulations to you, you were able to connect. So yak is the yeast artificial chromosome and it can carry up to 2000 kilo base pairs load, right? So which means it's a very big vector, right? But more than this is the human artificial chromosome, let me call it hack. These days, this is great for hacks, right? Computer hacks and life hacks and food hacks. So this is also another hack. This is human artificial chromosomes. Even human artificial chromosomes are used which can carry more genetic load than even 2000 kilo base pairs. So out of this list, probably the maximum load can be carried by the human artificial chromosomes. And BACs are somewhere here. BACs are the bacterial artificial chromosomes. So they can carry a little less than this, about up to 350 kilo base pairs, right? Alright, so that is a piece of information I would like you to assimilate. I would like you to know what are the different genetic loads. There are so many books available. There's one that even I have written. It's in the market from the MTG publications called Count on Me Biology. And I have given a comparison there of the different vectors, so you can even refer to that. So you must have a little bit of information of the different vectors in use, particularly those vectors which have been mentioned somewhere in NCERT. You need to know a little bit more about them, right? So the next question that I'm taking up is the 22nd question. 22nd question. 22nd question is, a gene carried by recombinant DNA is cloned when? Alright, so what is, and only 56.25% of you have done this correctly. However, this is one of the very simple conceptual questions straight from the NCERT. This doesn't even, ask you to be too careful about the text. It's kind of a very obvious fallout of the text. Let us see what exactly this means. Now, gene cloning would actually mean, I'm sure you know about cloning, right? Cloning means producing multiple identical copies of something, right? I can clone anything, right? I can clone, say, a figure, this is a figure I make and then I've cloned it here, right? So this is cloning while drawing. But in biology, yes, cloning can refer to the cloning of, we can clone cells. When a cell divides by mitosis to produce two identical cells, it is cell cloning, right? Then we can have organ cloning, and these days you even have organism cloning, right? You must have heard about the Dolly sheep, right? That is, in fact, Dolly was one of the first mammals to be cloned. But that is not to say that animal cloning is a very rare thing. The lower animals actually clone themselves very regularly in nature only. The animals which can carry out asexual reproductions are doing that all the time. And in plants also, during vegetative reproduction, cloning is very common, right? However, here we are talking of cloning at the level of the genes. So gene cloning means producing multiple copies of a gene, right? So, the gene cloning, when we talk about it with respect to the living cells, which means that suppose this is a vector, right? And this is the gene, the desired DNA that I need to be cloned, right? Which means I need multiple copies of this gene. So, what I can do is I can insert this vector with the desired gene, which means this is now a recombinant DNA, right, inside a host cell, right? This is a host cell. Now this could be a bacterium. Most likely this is a bacterium or a single cell organism like yeast, right? Now this bacterium, when it takes up this particular plasmid, the rDNA, now when this bacterium divides and forms multiple cells, 2 from 1 and then 4 from 2. Mitosis or binary fission, for example, leads to the formation of the different copies of the cell. So the cell divides to form more cells. So as this divides to form, more cells like itself, even this also divides, itself. It multiplies, it clones itself to be passed on to the different cells, right? So this will be passed on here also, and it may even form more than one copies, So if you recall studying it in the NCERT, the vectors that we are using for gene cloning have their independent origin of replication. The replication of this recombinant DNA is not dependent on the division of the cell. However, it may, so happen that when the cell divides, it divides not necessarily in a dependent way, not in a dependent way at all. In fact, it actually multiplies itself and is passed on to the different daughter cells. So when this divides further, more copies are formed. So this is what is gene cloning. So this is the amplification of the gene in vivo conditions. Now, what am I saying? In vivo. I'm using a Latin phrase in vivo, which means within the organisms, within the living organisms, right? So this means that within this living organism or the host cell, the gene is able to make multiple copies, because as the cell divides, more and more copies of the rDNA are also formed. There is one more method of gene amplification, or another method which can form multiple copies of a particular gene or a segment of DNA, and that is the polymerase chain reaction, right? Now that is a method which is in vitro, right? So let's contrast this. This is an in vivo method, whereas the PCR is actually an in vitro method. So that will help you in further to differentiate between the two processes of forming multiple copies of the gene. So here you can see this is an in vivo method within the living organism. That is an in vitro method. That takes place within a machine called the thermal cycler. This is a slow process, that's a fast process, right? So this is about question number 22. Yes. So the next question I'm taking up is question number 9, which has been marked correctly by about 16%, how many? 61%, sorry. 61% of you, and let's see what is question number 9 for you. Yes?
Muskan is saying
yes?
she got your co-authored book, Count on Me Biology, and it's really great.
It's really good?
Huh.
Thank you, Muskan. Well, it really feels nice when somebody appreciates your work. However, Muskan, if there are any fallbacks, any problems that you're facing with it, I will be so happy to get the feedback and we can make it even better for you, right?
And Ma'am, Rahul is asking, Ma'am, please explain YEP vectors.
Which
My
one?
EP vectors.
Yep, factors. Now, see, these are actually not in your syllabus. I would really propose that you not go into too much of these, So, in fact, it's never mentioned in the NCERT, never previously asked in the medical examination. Now, who's asking?
No, Rahul.
Now, Rahul, see, actually there are so many vectors on the board these days that if you actually go on learning all of them, have you heard of this thing called the RAM, random access memory? You know that a card or even a computer software has a certain amount of memory. If you keep putting in all sorts of information that is not going to help you in future, your memory will get full with that only. So I will definitely advise you humbly, I would request you not to overload your brain memory with unfair and not really asked for information, right? So do only as much as required, which means do what is in NCERT, do what has been asked in the previous medical examination. So it is very important for us to know what is relevant for us to do and what is not. We don't need to do a PhD on all the subjects and all the topics, right? So the next question, and that is 13, yes?
Number 9.
Number 9, yes, so okay, we haven't done number 9 as yet. Now, amplification of gene of interest by using DNA polymerase may go up to 0.1 million times, 1 million times, 1 billion times, and 1 trillion times. Well, this is a question, again, very, very straight from the NCERT, and I really don't know what made you answer this incorrectly. But Arjun, I would definitely ask the children once and tell me when they give me the response that what did they mark and why did they mark it? What did they think about when they were answering this question? Because this question is actually worded straight from the NCERT. Now, this is a question that has appeared previously in a medical examination. That is the reason why I put it up here for you. Now, there was a reason I put this question here for you. Let me explain that to you. Now, when you deal with the topic on the PCR, even from the NCERT, very simply from the NCERT, what you see is there is a piece of information given there that 2 raised to the power n is the number of copies of DNA obtained, right? Right? Where n equal to the number of thermal cycles. Now, I hope you know that the PCR, polymerase chain reaction, takes place in a machine which is called a thermal cycler, right? Now, this is a machine which actually alternates the temperature. It actually runs some temperature cycles, and according to that, the 3 processes of denaturation, annealing, and extension takes place, and this is how 1 copy of DNA forms 2, 2 form 4, 4 form 8, and so on. And therefore, there is an exponential increase in the number of copies of DNA, right? Now, in the NCERT, it's mentioned that if the number of cycles is 30, if n equal to 30, then you would form about a billion copies, right? So this is the exact information from NCERT. Now, theoretically speaking, if we increase the number of cycles from 30 onwards, you think theoretically it is not possible for us to make more than billion? Well, of course it is. Because if we increase the number of cycles more than 30, definitely we will get more than billion copies, right? And this actually figure can go on exponentially. But this is a classic example of a question which has been worded exactly by the paper setter from the NCERT, and he expects you to answer from the NCERT, right? So this has been, given as that. In fact, this is the answer given in all the textbooks also. So just take it from there, because practically it is very much not possible at times to run cycles more than that. Although theoretically it might be possible, right? But we will stick to NCERT for this particular answer, right? Because even if we had to say more than that, then we would have given an unending figure, not a definite figure. We would have said 1 trillion times. We could have said more than that, right? So this actually is looking for, again I'm talking to you about the psychology. Now, how would you understand? You may ask me here, ma'am, how would we understand whether the paper setter wants us to answer from NCERT or wants us to think a little bit more, right? Because if the N is increased, then even the number of copies of DNA would be more than 1 billion times, and then why not 1 trillion times is the answer, right? So here I would say, look at the options carefully. If the option is the fourth option is very, very specific, 1 trillion times, which means that the number of N has to be very specific also in this case. So when there is a very specific choice, then that actually means that the person is not looking at at you to think more about it. The person wants the exact figure, and the exact figures have to be from the NCERT, right? So I hope that satisfies you. And again, I did not make this question up. I did not frame this question. I have picked it up from one of the previous examinations, just to give you a sample of how sometimes an exact word or a phrase or a figure from NCERT might be asked of you, right? So sometimes you don't have to look at the exact phrase and connect unnecessarily, and sometimes you have to do that. So you have to really understand the psychology of the questions. You have to be a psychologist to crack NEET nicely, right? So I will help you be a good psychologist. You have to be a question psychologist, right? All right. So the next, I hope that satisfies you. So, there's something on your mind. If you still think there's something wrong about it, do come back to me. I will talk to you about it. We'll discuss, right? The next question that I'm picking up is 13. And let us see, 13 is a question. Okay, cDNA. Now children, cDNA, I am actually wondering why many of you marked it incorrectly, because this is something that you were supposed to know, right? Because cDNA, you're at the end of 12th, right? Which means you've finished up the entire syllabus once with your teachers in all the places. And that actually means, again, one more psychology point I'm telling you. So when you're actually reading a text, right? You're reading through a book, NCERT, paragraph, whatever. What we generally tend to do is skip small things, So you might have seen cDNA somewhere, written somewhere in NCERT. I'm sure it's there in the NCERT, right? So you must have seen it, but probably you thought this C is not very important, So that is one thing that I need to tell you, that sometimes small things can make a big difference, So if there is DNA written there and just before DNA there's a small c, don't think it's just there, just like that, or maybe a misprint, or maybe doesn't really mean much. It must think, you must think, okay, this must be some kind of DNA, not very important, but no. So if there is something written very specifically before DNA, you must try and analyze why is the C here, right? The small things definitely matter. This is this next psychology point that I'm telling you. So C here refers to the complementary DNA. So what is this complementary DNA, or cDNA as we call it? This is the DNA which is formed by reverse transcription from RNA, right? Now RNA is the genetic material in many of the viruses, particularly the particular virus that is in question now is a group called retroviruses. This also has, the entire group has RNA as a genetic material, and what they do is that from RNA they make a DNA copy with the help of the enzyme reverse transcriptase. So this particular enzyme is a very important enzyme for us. It helps us in genetic engineering also. It actually helps us to make genes which is not possible to be isolated as such. Now what am I saying? Let me tell you. Okay, first let me just tell you, this RNA which makes the cDNA copy by reverse transcriptase is termed as the cDNA, or the complementary DNA. It is complementary to this RNA. Now, when we are carrying out the procedures of genetic engineering, which means we need some specific genes that we desire. Suppose I need the insulin gene from the humans to be put into E. coli. Now, this was actually done, you know that, right? But just for a moment, imagine that this is a hypothetical situation, that I need a certain gene, but somehow I cannot extract it out from the cell. There are some problems that are coming up. Maybe I cannot find the correct restriction enzyme that can cut it in the right places, or whatever the reason be. I'm not able to isolate it from the cells. So there's one more thing I can do. what I can do is I can take the messenger RNA for insulin, right? And by reverse transcription, I can obtain a copy of DNA or a complementary DNA. You can also call it a copy DNA actually. So here, okay, complementary or copy. So these are the synonyms, right? So the copy or the complementary DNA obtained by RNA as such would help us to use that gene in the way we want to during the process of genetic engineering. So that is one of the ways in which we can obtain the desired genes in the process of genetic engineering. So I hope you are with me on that. And with that I go on to the next question, and that one is 13 is done, right? So 3 and 2. So we'll take up 3 and 2, both of which have a success rate of about 65%. Okay, so I'll take up 2 first. Right. Which of the following restriction enzymes produces blunt ends? Now, again, this is a very question which actually requires you to have some information, right? Well, you must not have heard about the ZHO1 enzyme, restriction enzyme, and EcoR5. You have obviously read about the EcoR1 that is given in the NCERT, but EcoR5 is one of the later finds, right? HIM3 and SAL1, definitely you have studied it somewhere in the NCERT. Now, the trick here is to have a list of the enzymes which give staggered cuts and blunt cuts, right? So you need to have, say, this is a small task you can do. Some of the important enzymes that give staggered cuts, write it on one side of the column and just know their names. You don't really need to know their restriction sequences, right? But I hope what is meant by giving a staggered cut and a blunt cut, I hope you know that. Or should I tell you? Okay, I might as well just tell you quickly here. For example, if we have the sequence 5' C T C G A G 3', and the other strand reads CTC GAG 3'. Now these are 2 strands of, there is double-stranded DNA with base pairing, and this sequence will be termed as a palindromic sequence, right? Why palindromic sequence? You know that palindromes are words or sentences that read the same forward and backward. I'm sure you must have heard of so many palindromes. I'll tell you about one. There's one sentence in English which is actually till now I think it is the longest palindromic sentence in English. Okay, I'll write it down for you. It's an interesting one. Was it a car or a cat I saw? Okay. So this is a sentence, right? But if you would need to rearrange the letters, but you can read it same backward and forward. Just like the word Aurora or Nitin. All these are palindromes. Eve, right? So all these are palindromes, right? Similarly, this is a palindrome. Now, what is palindromic DNA? Palindromic DNA is a double-stranded DNA which reads the same back and forth, right? But we will look at it from the direction of 5' to 3', right? Because conventionally, just like you generally read an English sentence from left to right, or an Urdu sentence from right to left, right? There is a convention in reading the sequences of DNA and RNA as well. We generally read these from 5' to 3'. So this particular strand goes 5' C T C G A G 3', and the other strand goes 5' C T C G A G 3'. So if you read it from 5' this side and 5' this side, it is the same, right? So this is a palindromic sequence. Now this is actually a restriction site for ZHO1, and this restriction enzyme actually cuts here. So any enzyme which does not cut exactly in the center of the palindromic sentence is going to palindromic DNA, is going to make a staggered cut, which means had this cut been here somewhere, here, exactly in the middle, 3 bases here, 3 bases here, then it would have been a blunt cut, no sticky ends, no unpaired bases, right? But this is what is a staggered cut. So it is going to cut here at this point between C and T and at this point between C and T. So this actually creates unpaired bases like this. So this is a staggered cut. And as I told you, blunt cuts are also made. Now there are some enzymes like SMA1 high 3, SMA1
pi 3, and as in this question we have E core 5. These are all enzymes that create blood cuts, right? So you'll have to do a little bit of work on this. Just create a short list for yourself, right? Maybe somewhere on the book or the notes that you have. Make 2 columns, enzymes with staggered cuts, enzymes with blunt cuts, and just keep looking in them. Which means, sometimes things which are to be learned, can be learned in a very effective way if you give it a repeated look, You can sit down with something to learn it, rote learning, but still, you may just remember it only for a day and then you again forget about it. So if there's something that you really need to keep in your mind, what is the best way to do it? The best way is to put up charts, maybe in your room, put up charts everywhere, writing down the information which is hard to learn. So whenever you pass by, you're just going to look at it, oh, ZXO1, that's a blunt cut. Oh, E core 5, that's also a blunt cut. So, that way when you're actually looking at the pieces of information all the time, slowly they will register into your mind in such a way that you can never forget them, right? So I used to do this, I used to put charts practically everywhere, on the walls, on the washroom door, and on the table somewhere. So wherever I look, there is a piece of information, right? So that's how, repeated impact. That is what is going to give you the real learning of the facts, right? Okay, so just try it and do let me know how it worked with you, right? So here the answer, as you can see, is the option 4. Now, for E core 5, I can tell you the sequence is GAT-ATC. So which means the cut is somewhere here. This is 5 prime, GAT-ATC 3 prime. Writing out the other end, GAT-ATC 3 prime. So this makes the double-stranded DNA, which is the restriction site for your E core 5. And you can see it is being cut right in the middle, so it is not going to produce a staggered cut or sticky ends or unpaired bases, right? So now we take up the question number 2. No, 2 is done. 3. So which of the following is not a component of downstream processing? Now we all know what is downstream processing. After we have obtained the recombinant protein through genetic engineering, yes, I hope you're all with me, which means there is a certain gene and we need the product from that gene, right? So what we have done is we have first isolated the gene, purified it, put it, first made a recombinant DNA maybe, or directly transferred the gene into the host, and somehow made the host express that particular gene. Now the product is there with you, but all the processes that take place between the point of time that you have the product in your hands commercially and the time that you market it, all the processes are termed as the downstream processing. So downstream processing actually deals with the purification of the product, right? Very important is purification, concentration of the product, right? And the third thing is stabilization of the product, right? So the product, and what is the product here? Most likely a recombinant protein that you have, got from a heterologous host. So to do this all, of course, there are a lot of things, into each one of these headings is a subheading. For example, purification can be done with the help of sedimentation, centrifugation, and so many different processes. Similarly, concentration also requires certain different processes to be, done. Stabilization is often done with the help of preservatives, buffers, you need to stabilize the pH and all that, right? So all these actually have a lot of processes going in hand. Now, among the following options, let us see what is the best choice here. Expression, separation, purification, preservation. Now, in light of the information that I have just given you, I think now you can tell the right answer, right? Well, you have been given the right answer, but now you can make, sense of the right answer. Why is the answer here 1? Now, expression, what does the word expression mean? A smile? Well, expression on my face is one thing, but what do you mean by the expression of DNA in terms of genetics, right? For us, expression means a certain, look on the face. But when we talk of expression in genetics, children, note it very carefully. Expression in genetics means that DNA forms RNA and RNA forms the required protein, which means you know that the genetic makeup is often referred to as the genotype, right? And the expression that it produces, or the effect that it produces, that can be observed, right, is actually through the proteins that it produces, and that is termed as the phenotype, which means when the genotype expresses out into the phenotype, that is what is expression. And how does that take place in genetics? It takes place when DNA first forms RNA and RNA forms the protein, right? So expression means formation of the protein, right? And we say the downstream processing are the processes which are applicable after we already have the protein with us, right? So expression is not a part of downstream processing. We already have the product. So to obtain this product, to obtain this protein, we have done this expression. The expression is needed here before the protein, not after it. So further on, now we will have to subject it to downstream processing. Yes? Now I hope you understand this, and you'll be more careful in future, because these are very small things. Obviously, this is not given very directly, but you need to understand the meaning of the word expression in terms of genetics to be able to answer this, right? Very simple question to look at. There's nothing out of NCERT in this, and yet, if you're really not careful about the words, there you go, right? Which means a very, very simple word like expression can actually put you in the loop, right? And it can actually, make you 5 marks or 4 marks behind what you actually deserve, right? So the next question that is coming up is 23 and 19. So let's take up 19 first. So children, are you going on strong? Are you enjoying this session? If there's anything that you want more, do keep asking questions. Let's be more interactive. I'm waiting for your responses, okay? Yes, anything that you need to discuss, biology or no biology. So even psychology or even methods of prep. I'm sure Kapil sir is doing that all the time with you, and he's so good at it that you probably don't have any questions anymore for that. Okay, so question number 19 I'm taking up first. That is, following are the essential requirements for recombination. Yes, this is a very important question, and this is something that I would really need to explain to you, because it's not very well explained in the NCERT anywhere. So this and again the question arises, why did I put this question up to you? This question has also been asked in some previous medical examinations. In fact, a variant of this question was asked in another exam. So about 2 or 3 times this kind of question has been asked before. So I really thought that it is important for you to know this. Now let us understand. Yes.
Shivanshu is asking.
Yeah.
Wait.
Shivanshu. Yes, Shivanshu, what is your question?
Let me scroll down. Ma'am, expression take place after or before downstream process?
Before. When expression takes place, then only we will have the protein, right? Because transcription and translation take place, and then only we get the protein. And downstream processing takes place when we have the protein with us, and we need to purify it and market it. Just like Shivanshu, suppose I need to market insulin. You must know that now there are insulin pens available, Earlier it used to be so painful. There were long needle syringes and you need to stick them inside. But now the diabetics have it really easy, because they have those pens, with that thin needle, and they can actually prick themselves and give themselves an insulin shot. However, how does that insulin, that human insulin, come to you? You have read it in your biotechnology chapter 2, the second part of the biotechnology, that the Eli Lilly and company and the pharma companies are actually making this human insulin with the help of the E. coli cells through genetic engineering, right? So to get that insulin, you need expression, which means RNA has to be formed, then protein has to be formed, and then you get the insulin, because insulin is a protein, right? But after you have that insulin from the E. coli, if you have taken that, But you cannot, immediately market it as such. You will first need to purify it so that there is no contamination in it. Then you will concentrate in the right type of, concentration required for the patients. Then you will stabilize it. You will add some pH buffers so that, when it is transported or when it is stored, then it is, it doesn't lose its efficacy, right? It remains efficient. So all these processes that take place between the product and marketing, and ultimately when it reaches the customer, that is downstream processing. So expression comes before downstream processing, right, Shivanshu? So I will now move over to the next question, and that is the following are the essential requirements for recombination. There's another question in the paper which is similar to this one. It requires you to know what exactly the recombination event requires. It requires single-stranded DNA, it requires DNA ligase, it requires DNA polymerase 1, and all of these. And the answer is all of these. So let's try to understand this. Yes, we need Alright. Now first things first, what do you know recombination as? What is recombination? Recombination means combining of the DNA from two different sources to form new combinations, right? Now recombination is not an event that takes place only once in a while. It's very much obvious that recombination takes place in nature also. When does it take place? Recombination takes place when crossing over takes place. Yes? So you have studied about it in genetics, right? So when crossing over takes place, recombination is taking place. When a virus is integrating itself into the bacterial genome, then also recombination is taking place. When a gene that we have put inside a cell also goes to combine with the chromosome or the DNA of that organism, then also the recombination is taking place. So recombination is responsible for creating new combinations in nature, as in case of the crossing over. It's also required, when viruses have to integrate themselves. I know you have read about the lytic cycles, lysogenic cycles. So we call the lysogenic cycle of the virus. In the lysogenic cycle, the virus DNA goes and joins the bacterial chromosome and it is called the prophage. So how does it do that? That is also a recombination event. So recombination taking place is in the nature also, and recombination takes place when we are trying to put a certain gene into a different host, a new heterologous host also. However, what exactly is this recombination procedure? How does it take place? Now, we must understand that it generally takes place when one of the DNAs at least is in a single-stranded form. Now, how does that happen? Suppose we have a DNA, a double helix like this, right? And we have another DNA which is single-stranded, right? Now, what happens is there is a protein, a certain protein which is called the REC-A protein. Now, when I am talking about the REC-A protein, I am also answering another question in the, in this particular question paper. So be very careful about it. So there is a very important protein called the REC-A protein. That's variants of which are actually found in all the living organisms and is very important for the recombination event. Now this REC-A protein first binds to the single-stranded DNA, right? Now, this has another site. In fact, it has two more sites. One site is for ATP because it needs energy to carry out this process of recombination. It actually acts as an ATPase enzyme also, right? And there is one more site through which it can attach to a double-stranded DNA. But which double-stranded DNA? It will not go and attach to any double-stranded DNA. It will actually, have you seen the shunting process? Have you seen the invigilation? Sometimes, this would actually go over this entire DNA to find a place where this particular single-stranded DNA is complementary to it. Now, this obviously the DNA that we are talking about here is a very long DNA. This probably is the DNA of the bacterium or another chromosome if it is a crossing over event. So this REC-A protein looks for a stretch of DNA here which is complementary to this single strand. So suppose when it goes over, it finds a stretch, say from here to here. This is somehow complementary to this, which means very much similar, but not exactly, right? Now what it does is it goes and attaches to this part, right? So from here it attaches to this part, and what happens then is a process called branch migration. What happens is, are you with me? So what happens is branch migration. So what happens is that it causes this part to, break the hydrogen bonds, and this DNA to form a link with it like this. So the other DNA is, is removed like this, right? The other strand is removed. So this particular stretch goes to form a, duplex with the other strand of the DNA. But this particular duplex which has been now formed is actually a heteroduplex. Why a heteroduplex? Because they have similarities but they're not exact, which means there are some places where they're not exact. And this is the reason why a new combination is going to be coming up, right? So further on, these the differences in the code are actually filled up by using the DNA polymerases, and the gaps, this particular end of the single-stranded DNA has to be joined to the rest of the DNA through the DNA ligase. So all these come into picture during the recombination event. The Rec A protein is required, and it only associates with the single-stranded DNA, never with the double-stranded DNA, which means whenever we put a gene inside, that gene can never go and join the internal chromosome in a double-stranded form. It has to come in the single-stranded form first, and the Rec A protein first combines with that single-stranded DNA, then it looks for a complementary place on the duplex DNA, then it induces branch migration through which one of the original strands is replaced by this particular strand, and the ends are sealed, and the parts which are mismatching, the mismatched parts are then removed and the gaps filled by DNA polymerase 1. So that means all these are actually required for the process of recombination. Now, the details of recombination are not in our syllabus, but by giving you the simple process, now you will be able to appreciate what is exactly the role of these enzymes, right? So the next question that I am taking up is 23.
We have two doubts from the
Yes,
students.
let me first take the doubts.
Aman is asking
Yes.
how protein binds to DNA,
Yes.
and Muskan is asking in human lymphocytes why retrovirus is used for introducing foreign segment of DNA.
Okay, let me first come to the first question. The
Aman.
first one was Aman's. Aman question is, Aman, what's your question?
How
Once
protein
again?
binds to DNA?
Protein DNA binding. All right. A very, very intelligent question, Aman. I must say, I must compliment you for your inquisitiveness. Now, if you really want to study this in greater detail, I guess you should, actually read up a little more about this. However, it's not in your syllabus, but I will definitely tell you a little bit about it. Now, there are places on the DNA with certain specific base sequences which are recognized by certain proteins. Not all proteins can go and bind to the DNA. There are so many proteins lying about in the cell and even inside the nucleus. But can all those proteins go and bind to the DNA? No, they cannot. Right? What is the reason? The reason is that the proteins have a certain shape which recognize a certain sequence on the DNA. And these parts of the DNA are actually called DNA motifs. What are they called? DNA motifs. Right? So there are certain sequences on the DNA which can be recognized by certain proteins. So these specific proteins are called DNA binding proteins. And you have studied about certain DNA binding proteins. I'm sure you have. And again, link the information to the second chapter of genetics. You have actually learned about the repressor. Recall the operon model? Lac operon, and I don't know if you've done the tryptophan operon, but even if you recall the lac operon, you must recall that there is a repressor protein which can go and bind to the operator region. So how does that protein go and join only to that operator region? Because there is a sequence in the operator region which can be termed as a DNA motif, which is recognized by that particular protein, and therefore it can go and bind. Right? There's another question in this which is somewhat related to it. I will come to it. It's called that zinc finger analysis question. So that is also very related to this concept. Zinc finger is also a protein which can bind to certain parts of DNA to either switch the genes on or switch them off. So So that is again an example of a protein binding to DNA, and that is done specifically because of certain regions being recognized in the form of DNA motifs, right? Okay, and as you can see here, then the Reg A protein is also a DNA binding protein, right? Also the enzymes polymerases. Obviously, the polymerases and the the RNA polymerases, the DNA polymerases, they all are DNA binding proteins, because otherwise how will they go and polymerize the DNA or the RNA, right? So recall, now this is what I call thinking deep. Yes, so I must compliment. This is what I call thinking deep. What kind of proteins can join to DNA and how they can do that? And have I studied about some DNA binding proteins? If I ask you, Aman, have you studied about DNA binding proteins? You will say, no ma'am, there is no such topic in the NCERT. But if you really think deeply, if you've actually read about so many proteins which can go and bind with DNA. DNA polymerase, RNA polymerase 1, 2, 3, right? And Reg A protein, as I'm telling you. So you've already read about them. The only thing is to think sensitively, right? And be aware of what you've already read. Very good. So that was Aman's question. And what is Muskan's question? Muskan was asking me about, I think, lymphocytes.
In human lymphocytes, why retrovirus is used for introducing foreign segment of DNA.
Alright, that's a good question, Muskan. Now, your question to me is, if this is the host, right? This is the host. This is the cell into which we have to deliver the gene, right? And this is the vector that we are using. Right? So actually the question that you're asking me is that what is the relation between the host and the vector? Which means, how do we decide which particular vector can be used for which particular host? Right? Now this is a very, so to speak, it's a simple question, but let me talk to you about an analogy, right? Suppose there is a Bengali family. Now I'm just saying that out of the blue. Could be any other family, but suppose there's a Bengali family who needs to be who needs to be sold something. There's a company who's selling some items, and the company needs to send a salesperson, right, to sell that item, right? And they know that this particular block has many Bengali families. So what do you think that the company would think about before sending the salesperson? The choice of the salesperson will depend on which factor? The salesperson needs to know Bengali, right? So if the salesperson knows Bengali, he will surely go and connect with the people faster, would be accepted by the people faster, and would be able to sell the product, right? Similarly, when we have a host, suppose this is a host and I have to deliver a gene inside the host, how will I choose? Okay, which vector do I choose? This one, this one, this one, this one. What will make me decide? Somehow or the other, there has to be a connect between the vector and the host, right? So if we can use a vector which either is accepted by or recognized by the host cell willfully, voluntarily, or somehow that vector has a way of entering forcefully. Because not every say pathogen can enter every host, for example, right? For example, if I am going to ask you, say there is this tobacco mosaic virus, right? If I drop the tobacco mosaic virus on your head, will the tobacco mosaic virus be able to enter human cells, your cells? No, because there has to be some degree of relationship, right? The virus has to recognize your membrane proteins. It has to enter your membrane proteins, but it won't be able to, because it is actually a pathogen only for the tobacco cells or the tomato cells or a specific group of hosts. Similarly, the vector has to be either accepted voluntarily or has to know a way to enter forcefully. Now, this is the host cell, the lymphocyte, and the retrovirus, the human T lymphotropic viruses. You have heard about them, I'm sure you have. You have heard about the AIIMS virus, that's HTLV3, right? Human T lymphotropic virus, right? So all these retroviruses are actually the natural pathogens of these cells. You know that these retroviruses, the HTLV series of retroviruses are they normally can infect our lymphocytes, so they know a way to enter the lymphocytes. That's why we are selecting them. To give you another example, there was a scientist called Crystal who did the gene therapy for cystic fibrosis disease. That's also a genetic disease that affects different parts of our body, including the pancreas, but the most prominent effect is here in the trachea and the bronchi and the respiratory tract. The cells, they make a lot of mucus and the person is always choked on mucus, So what he did was, okay, so if there is a gene to be delivered into the cells of the respiratory tract, what kind of vector would you choose? A vector that knows how to enter the cells of your trachea or the bronchi, right? So that is your common cold virus. So they made use of the common cold virus. Obviously, these viruses have to be disarmed or attenuated, if you know the word correctly. You must know this word. Attenuated means disarming. Disarming means taking away the guns, right? To take away the gun, to take up the weapons of something. Which means we have to make these viruses, these pathogens harmless. We have to remove the genes which can cause harm to the host. We have to use only the rest of the parts, right? So we have to use these viruses only after disarming them, right? So that's important for us. Attenuated pathogens can be used, but you have to use a natural pathogen, So if your host is lymphocyte, then you will use HTLV because HTLV normally infects the lymphocyte. So it knows a way how to enter that particular cell. That is the choice of the vector you have to do, right? Obviously, there are some direct methods also. We can, bombard it with some, gene guns are used, for example. Microinjection is done. Laser-mediated transfer is done. PEG-mediated, PEG-mediated transfers are done. Some direct methods are also there. But many times in certain cases, we have to make the choice very carefully, because this particular virus knows not only a way to enter the cell, but also it knows a way to integrate its genome into the genome of the host, which is what we want. So that's a very good thing, right? So very good question again, Muskan. And again, it shows that you are thinking deeply and you're thinking beyond just the statements in the NCERT. I'm very proud of you and very good. Keep doing that. If that is your spirit of inquiry, I'm sure you will succeed. Bye, best wishes. The next one is question number 23. Restriction endonucleases are present in mammalian cells for degradation of DNA when the cell dies. Again, this question is directly from the NCERT. If you recall, the opening statement where it starts with the restriction endonucleases is that in the 1960s, two enzymes were discovered, right? Okay, let me write them down for you. I'm a little thirsty, and if you also have a draught of water quickly. You might ask me, why am I drinking from this, why not a glass or something else? Well, I would like to share this small information with you. In the ancient texts in India, it's been mentioned that when we drink water out of a curved vessel, it actually lessens the surface area. That's some physics. I'm not very conversant with physics, but actually the water which is drunk out of a curved vessel will affect your body very positively. But if you drink out of a straight vessel, it will not have that good effect, that positive effect on your body. So it's always better to keep your water for some time in a curved vessel and then drink it. That's a big science. So our Shastras, our Vedas have such big science, You must appreciate that. So, so I was talking about the opening statement that in the 1960s, two enzymes were discovered. One was methylase and the other one was restriction endonuclease. And there's a statement also made there, if I recall correctly, that both these are a part of the defense system of the bacteria or prokaryotes, whatever the word mentioned there, right? So it is actually right from there, right? So there was no reason for you to make a mistake here if you had studied your NCERT properly. But let me just tell you a little bit more about it. That how exactly does this defense system work? Well, we have a defense system, right? You know that whenever a pathogen tries to enter our body, there are different lines, at least three, I think if I recall correctly, there are 3 lines of defense, right? Our skin is the first defense, and then we have so many other things, including the beta lymphocytes, the T lymphocytes, which protect us from all these germs. Now, the bacteria and the prokaryotes are single-celled primitive organisms which do not have the very sophisticated defense systems that we have in our body. But they do definitely have some defense system against their pathogens. Now, what are the pathogens of the bacteria? The bacteria are pathogenized or infected by the viruses. We are, of course, pathogenized by viruses, bacteria, fungi, all sorts of microbes and germs. But for bacteria, their pathogens are viruses. So they need to have a defense against the viruses. Now, let me just draw it a little figuratively so that you can understand it well. Suppose this is the bacterium and this is a virus. This is the virus DNA, and you must have studied this, how viruses put their genomes inside. They insert their DNA inside, much like it's giving it an injection, a shot. So when the DNA of the virus is inside the bacterium, then it tries to take over the entire machinery and form its own multiple copies. It uses the entire bacterium to make more copies of itself, right? And ultimately, many of the viruses, which are live viruses, may actually, burst the cell and, escape out. The copies may escape out. So what happens is the bacteria need to have a defense against this. So most, I wouldn't say all bacteria, because probably the researches are not that complete till now, but at least more than 25% of the bacteria studied till date do have these restriction endonucleases that we're talking about. So they make certain restriction endonucleases which can recognize some sequences, certain palindromic sequences found on the virus, so that when the viral DNA enters, this can just go there and quickly cut it up. So once the viral DNA has been cut up, it cannot do any harm to the bacteria. That's how it works. Now, what are the methylases for? The methylases are, what are they doing? They're actually methylating the same sequence that the REs are cutting here. Now, suppose there's a sequence called GAATTC, as you know, ECOR1 recognizes, right? Now, suppose that the same sequence is present here also, GAATTC. Right, GAA, there has to be one word, A, right, like this. So, same sequence present here also, but it is cutting up only the sequence found in the viral DNA, not on its own DNA. Why? Because its own sequence is protected by methylation, by adding methyl groups. So, when methyl groups are added to the bases, the endonucleases, any type of endonucleases in fact, cannot recognize that part of DNA and cannot cut it up, right? So they protect their own restriction sites by methylating them, but they can cut up the restriction sites, wherever they find the restriction sites on the viral DNA as a part of their defense system. This is how the defense system works. Pretty intelligent, right? Even the little ones, the prokaryotes. So to speak, these people don't have those nervous systems and very high intelligence. But still see how they're managing so well on their own. Right? And the next question I'm taking up now, unless you have it out, is 18. Yeah.
That Shivanshu is asking, why restriction endonuclear? Endonuclear is not found in human cells.
Okay. Now, as I was telling about to you, Shivanshu, restriction enzymes are mostly found in the prokaryotes. In many of the prokaryotes studied, but not in all. In many archaebacteria, that's of course a part of the prokaryotic part only. Then very few eukaryotes. Very rarely it's found in the eukaryotes, and that too mostly the unicellular ones. But not in the higher organisms like humans, like higher plants. Why is that? Because multicellular eukaryotic organisms like us have very sophisticated lines of defense, as I was telling you in the initial part. We have a very sophisticated system of defense. If you have studied the chapter on immune reaction, immunology in human health and disease, I think it's somewhere there. So you must have read there that we have 3 different lines of defense. First line of defense is the skin. And now first is to block the entry of the pathogen within the body. What to talk about the entry into the cell. We will talk about the entry into the cell at a later stage. So the first line of defense first works. If that fails, then second line of defense works. And then that fails, then the third line of defense. So which means this particular RE is actually a cellular defense, which means a defense of the cell, right, against the DNA that is entering from outside. But why should we wait, you know, as multicellular organisms, why would we wait for the bacterium first to go inside the body and then only, or when it is about to enter the cell, then only we exercise the immune response? No. So our immune response starts right from the exterior of the body only. So our defense systems are much more sophisticated. We don't need that primitive kind of defense. It's a primitive type of cellular defense, right? Well, again, but who is this? Shivanshu, na? Shivanshu, again a deep question. But I would like you to link this up again. Link this piece of information that you have asked me to a section in the biotechnology part 2. You have studied about a system of cellular defense in eukaryotes also, and that is the interference RNA. So we also have a kind of cellular defense like the bacteria have here, but that's in another form, and that's called the interference RNA technology. And we are using it, in fact, in genetic engineering to deliver genes into certain eukaryotic cells. And you have studied about the nematode, the nematode case, right? So similarly, the type of defense that this RE is exercising is actually a primitive type of defense, which is found mostly in the prokaryotes. Eukaryotes also have lots of defenses, you know, first, second, third lines of defenses, and then these intracellular defenses. The intracellular defense that we have is the interference RNA and not the restriction enzyme, right? I hope that satisfies you. So we are now going on to the next question, and that is, we have done 23 and 19, no?
Yes, ma'am.
No.
I guess it's 18.
Now it's 18, okay.
Yes, ma'am.
So the 18th question says, Rec-A protein binds to. I think I've answered that. Now you can answer me once again. The Rec-A protein binds to the single-stranded DNA. So
Ma'am, Muskan is asking how nomenclature of virus is done.
How?
Nomenclature viruses is done. It gets difficult to remember their name.
I
Please
know.
explain.
I know, Muskan, that's very right of you. In fact, the names of the viruses are like names of some Sten guns. You know, there was a gun called AK-47. So when I talk about a virus which is M13 or 5X174, it actually makes me think maybe it is the name of a gun or a missile or something like that. But okay, to answer the first question that you have asked me, how are they named? Actually, there is no particular reason here. Mostly it is to do with sometimes the host that they're infecting, sometimes the number of capsomeres that they have, sometimes the kind of nucleic acids they have. Jibs, there was a scientist called Jibs who actually gave a 4-part code for each virus. That's called the Jibs code for the virus. However, that's not under the purview of our discussion today. But there's no very specific way to name the viruses. As you can see, tobacco mosaic virus is named after the host that it is infecting and the type of symptom that it produces, right? And this M13, not AK-47, I'm sorry, M13. So these are, probably to do with some first letter of the host. So there's no very specific way to name the viruses, Muskan. I'm really sorry I can't help you in that, but you'll have to memorize the names as such, right? So question 18 says, question 18 is done, right? I hope you don't have a problem with that because we just discussed the role of Rec-A protein, how it binds to single-stranded DNA, and then it induces the branch migration. So again, let me recall that with you. Rec-A protein can actually bind to single-stranded DNA on one side. It can also bind to complementary double-stranded DNA on the other side. It can also bind to ATP. So it can actually bind to 3 things at the same time. Okay, so the next question that I am taking up is 6, and the 6th question is, okay, let me turn the page. Alright, polyethylene glycol method is used for biodiesel production, seedless fruit production, energy production from sewage and gene transfer without a vector.
Which question?
This is question 6.
Okay.
Six. Arjun, can we have 6 on the board?
Yes, I will.
Alright, so PEG, as I was indicating a few moments ago, that some methods of gene transfer into a host are called direct methods, which means we are just directly putting the desired gene inside the host. Now, there is something I think which I would like to elaborate a little bit more about it. There is an analogy that I have written in my book about this, and it's I think it's very nice. And if I use that analogy to make you understand how a host cell would, accept, or how a DNA would actually enter a new host, right? Now, I will take up 3 cases. The first case is, suppose I am a lady inside the house and somebody knocks at the door, right? I will go to check and see, maybe through that peep door, and see who it is, and then only if I know that person, then I'll open the door. Now, suppose I, see through the peephole and I see, oh, this is the regular wash person who comes to my place. So it's okay, I know him. I'll open the door and the person enters it. Which means the first method of the entry of the gene inside the host is when the host recognizes the gene, right? And that is what exactly happens when, in the case of Griffith's experiment. The R strain bacterium picked up the S strain bacterial gene because it was related to it, right? Which means that the membrane receptors are recognizing that part of DNA as somewhat related to itself. So that is voluntary gene acceptance, right? Now, the second method. Now, somebody knocks at the door, I go to the door and look through the peephole, and I see this person looks familiar. I know this person. But that person is actually somebody bad but holding a mask of somebody I know, which means the person is in disguise, right? So I feel that I know this person, but that person is actually in disguise. So I open the door voluntarily, but that person then enters, and then of course something bad can happen, right? So that is the case when I send the gene through a vector, maybe through a pathogen, right? In fact, rather through a vector, right? So when I send it through a vector, I'm sending it through in a disguise, or maybe disguised in a liposome, for example. Suppose I have this lipid globule inside which I put the DNA, and the lipid is very similar to the lipid matrix, and then it just enters inside in disguise. So that's like entering in disguise, right? Now there are the third set of methods, which is the direct gene transfer, which means forced entry, which means when I go, somebody is knocking, when I go to look at the door, I see, oh my god, it's a dacoit, it's a thief. What do I do? So that I don't open the door, but the person outside, the dacoit, he just shoots a gun and opens the door and through the lock, and God forbid, may that never happen. But that's a forceful entry. That's a forced entry, right? Similarly, forced entries can be made through the pathogens, the pathogens that infect the cells. So we can use the viruses, for example, as the vectors for that forced entry, or we can use the gene guns or micro-injections, macro-injections, laser-mediated, and so on and so forth. And one such method of direct gene transfer is the PEG-mediated transfer. Direct transfer methods, lots of them are available. So PEG-mediated polyethylene glycol. Now this is a chemical which, when used along with a divalent cation like calcium, can actually cause temporary destabilization of the membrane in such a way as to create maybe pores, or we can say it makes it more permeable, right? So it makes the membrane permeable so that it can take up the DNA, right, easily. So this is a method used for direct gene transfer. It's used for the plant cells also, the animal cells also, but the thing with it is that it has to be used on the naked protoplasts, which means we cannot have a cell wall over it. It will not work when the cell wall is present. So cell wall has to be removed if it's a case of the plant cells, right? And very importantly, this PEG is also used for fusing two cells, two protoplasts. So it is actually also used for another method, which is another process rather, which is called protoplast fusion, right? So PEG is a substance that you need to remember, right? So the next question that I am taking up is, this one was 6, right? So I take up 8. The restriction enzymes used in recombinant DNA technology that make staggered cuts in DNA leaving sticky ends are all of these, right? So this is again a piece of information that I told you must have, and staggered cuts are made by all of these. So I will assume that now you will make those two columns, because again, both these questions that have been put up from the restriction enzymes have been for the sole purpose to make you understand that these have actually appeared in the previous examinations. So you need to give them that adequate importance, right? Next question I'm taking up is 24 and first. In fact, let me take up the first one first. Which of the following is not required for any of the techniques of DNA fingerprinting available at present? Now what is DNA fingerprinting? We need to talk a little bit about DNA fingerprinting. Now, we know that all of us have a major portion of our DNA which is termed as a junk DNA, right? In fact, suppose, let me talk about the human DNA, because as of now we're doing the DNA fingerprinting only for the humans. There has been no reason to do it for the animals or the plants for that matter. So human DNA, in fact most of the higher plants and animals DNA for that matter it's correct, this information is correct for that. Human DNA has about 98 to 99% junk or redundant, which means DNA which is not coding for, proteins, functional proteins, right? And only 1 to 2% is functional, which means coding for proteins. Now, you might ask me what might be the reason for that? Why are we walking dustbins for DNA? Well, it does look like that initially, but there has to be a reason. Nature has its own reasons. Nothing really can be junk. And in fact, now the scientists, as they are researching more and more, they're finding out that the junk DNA is not really junk. It must have some reason why it is still present in us, right? So, however, this so-called junk DNA has some parts of DNA which are termed as repetitive sequences, repetitive DNA, right? And this also, the repetitive sequence is also called the, I may call them as, there are different types of repetitive sequences. So they can be termed according to the types as satellite DNA, right? So this is what is the basis. Now the number of repetitions of the DNA is what actually is important in DNA fingerprinting, because we call them VNTRs, variable number of tandem repeats. So what is the basic process here? So in this basic process, we need to know that we have a variable number of tandem repeats repeated a different number of times, and when this happens, we need to actually cleave that particular part of the VNTR and find it through a probe. So, if I actually go into explain the entire process, that will really take some time, so I'll refrain from doing that. But let me just summarize it by saying, in this particular process, I am cutting up the DNA with the help of some specific restriction enzymes. So restriction cleavage is important there. And then I'm separating them using electrophoresis. And after that, I'm blotting them onto a nitrocellulose membrane through the process of southern blotting. Then I'm using a probe to identify a certain VNTR and then highlight it, right? And make those bands. So here, which is that particular thing which is not important? Now, polymerase chain reaction is important. Why? Because PCR is used to amplify the DNA sample, right? So to amplify the DNA sample, we would need the PCR, because suppose at the crime site we have this very small piece of blood available. So unless we amplify it, we will not be able to carry out fingerprinting and identify the criminal in that particular case. Then restriction enzymes, as I told you, we need to cut the DNA at specific places using the restriction enzyme. So that's also important for DNA fingerprinting. DNA hybridization, why is this needed? Because we use the probe, which may itself be a DNA sequence. So unless the DNA hybridizes with another known DNA, we will not be able to find out the bands. So the only thing that comes out from here is zinc finger analysis. You could have marked this because it's not given in NCERT. This particular option 2 is not given in NCERT. So that was a sure choice. All the rest are there. Right. So what is zinc finger? So I was telling you earlier also, zinc finger actually is a protein. It's a DNA binding protein, right? DNA binding protein that plays an important role in regulation, which means in switching on and off of the genes. In fact, sometimes it is known to start the expression of, activate certain cancer-causing genes like proto-oncogenes also, right? So you could read up a little bit about it if you're interested, but this is what you actually need to know, that zinc fingers are proteins which are DNA binding proteins and have a role to play in regulation of genes. And of course, they do find an application in genetic engineering and in genetics also for different purposes like switching in on and off of the genes and so on, right? Next, I'm taking up the question 24. That's the last part one question. Let me come to it. Electroporation procedure involves fast passage of food. Now, and I'll come straight to the correct option here. Making transient pores in the cell membrane to introduce gene constructs. Now, what is electroporation? This is something given in the NCERT also. And when we subject the cells, the protoplasts, to transient, which means short-lived or very short-timed voltages, currents, then what happens is, that membrane is a polarized structure. The inner and outer parts of the membranes may have some polarization. Sometimes polarized, sometimes depolarization. If you connect this with your nervous transmission, I'm sure you'll be able to connect with it. So when we apply this electric current, somehow the displacement of charges takes place in such a way that the membrane becomes, it gets some, temporary pores. So that helps for it to pick up the DNA from outside. Just like PEG may, make the membrane more permeable to pick up the DNA. Similarly, giving electrical currents may also serve the same purpose to create pores inside the membrane through which the DNA can be picked up. Right. So the next question that I'm taking up is 20th. Let's see what made you. Well, the 20th question was done correctly by 81% of you. And let's see if there is any problem with it. 20th question, ideal host for the amplification of DNA molecules is bacteria. Why? Why bacteria? Now see, an ideal host is one which is easy to replicate, right? Now bacteria actually multiply once, for example, if you talk about E. coli, it replicates once every 20 minutes. So whatever gene we put inside, it will be cloned very quickly, right? And easy to replicate, easy to culture. So all these things are seen when we talk about the host. However, if there are certain vectors, certain recombinant DNAs which would not be accepted by a certain bacterium, we might have to look for different hosts. For example, yeasts can act as hosts for certain genes, certain recombinant DNAs. Sometimes even human cells can act as hosts, but most commonly it is the bacteria because they're easy to replicate, they divide very fast, easy to culture in the laboratories, so that they make very, very favorite as candidates for the hosts, right? Now 14th question says, restriction endonuclease, an enzyme used in genetic engineering, is employed for cutting only double-stranded DNA. Now this is something that we've already covered. Restriction enzymes always cut the palindromic sequences which are always double-stranded. So that I think is beyond any doubt or confusion. We can, I think, move on directly after this. 21. Let us see. 21 has been marked correctly by 82% of you. And this says, a piece of nucleic acid used to find a gene by forming a hybrid with it is called. Now, probes are like, I call them like police dogs, just like police dogs go and sniff out the criminals. Similarly, the probe can either be a molecule of DNA or RNA, but it has to be single-stranded because it has to go and bind, right, to another DNA or RNA. And it can even be a protein. Which protein? DNA binding protein once again. So it needs to recognize some motif on the DNA, right? So these probes are helping us to recognize certain sequences on the DNA. So here we are talking about a piece of nucleic acid which can either be DNA or RNA. So both of them can act as probes, right? The next one is 10, 12, 17.
What you saying? Can you repeat 20 questions again?
20 question. 20. Which
Ideal
was
host of
ideal host. Okay. Now Shivanshu, an ideal host actually depends on our purpose for gene introduction. Suppose we want to obtain a certain product from a host, right? What would we like? That host cell should be able to replicate fast in the culture. We shouldn't have a cell which takes 1 day to, one cell making 2 in 1 day. That's too long for us. So if it's a particular cell which is dividing very quickly, it is easy to culture in the laboratories, that's an ideal host, right? And bacteria, and particularly E. coli, Escherichia coli, is a very, very favorite host because it takes up the, it is actually very, very receptive to a large number of vectors, right? So its acceptability is also very important, right? So all these things make an ideal host. But as I was telling you, sometimes where need be, yeast cells or the other eukaryotic cells may also act as hosts. For example, for the platelet-derived growth factor, we are using yeast. For the hepatitis B vaccine, we are using yeast as the host. But again, yeasts are also single-celled, relatively easy to culture. So there are a lot of considerations when you're talking about hosts. But most favorite hosts are the bacteria because of the ease with which we can replicate them, right? Then 20. Which one is now there? Which one we have to do is 14?
No, 14 is done.
14 is done. No, 10, 12, 17.
10,
Yeah.
12, 17.
So we have talked about 10, 12, and 17. Okay, let me take up 17. It's right here on my page. Plasmid's replication is autonomous, depends on the genomic DNA, needs an initiator, does not replicate. This is straight from the NCERT, children. You know that a good vector is one which has an autonomous replication. It has its own origin of replication, right? So if it is dependent on the host cell, then it defeats the purpose, right? So that's pretty straightforward. 12 says, enzymes commonly used in genetic engineering are. Now this is a question again from one of the previous exams, and why I wanted to put this here up was that sometimes the options given to you are all correct. Then what do? You actually look for the most appropriate in that case, right? So in fact, many times if you notice, somewhere up there on the question paper it is written, take the most appropriate choice. They usually do not write take the correct choice. So sometimes more than one options may be correct, but in fact sometimes all the options may be correct. But we have to look for the most appropriate out of them. Now if you see this here, endonucleases is actually a big class of nucleases, and endonucleases is a big class of enzymes which cut the DNA from the middle. But even restriction endonucleases are endonucleases. So if you look at this choice, it actually is a correct thing. I mean, we cannot say that endonucleases and ligases are not used in genetic engineering. But why have we marked the option 4 as the correct option? And the reason for that is that's the most specific choice, right? If I had all of the above somewhere, right, then I could have taken that also, because endonucleases, even polymerases are used during the recombination events. Ligases are definitely used to join the ends of DNA, and endonucleases are definitely used. So actually speaking, all these enzymes are used in genetic engineering, right? But the most specific choice, if you have to take one, is the last one because it's very, very specific, right? Then we take up the question number 10, and that is, which of the following is not correct for the organism and its cell wall degrading enzyme? So for that we need to know which particular material is making up the cell wall for bacteria. Bacterial walls are made up of murine, peptidoglycan. This is broken down by lysozyme. Plant cell walls are made up by cellulose, so cellulases can break them down. Fungi have chitinous walls, so chitinase works there. And what about algae? Even algae have cellulosic walls, most of them, right? So here obviously the choice should have been, the correct matching should have been cellulases, not methylases. What is the function of methylase? We talked about it. It's part of the defense of the prokaryotes, right? Along with the restriction endonucleases. So methylase is not the correct answer, right? Then we will talk about the last question. We are at the last question of the question paper, marked 90% correctly by 90%, and that's the 5th question. So it looks 5th question should be the easiest question. So introduction of tDNA into plants involves infection of the plant by agrobacterium tumefaciens. It actually is probably the most easy question on the paper. Very direct, fact-based, no twisting, no traps, right? So definitely agrobacterium tumefaciens is a bacterium which causes a disease called crown gall mostly in the dicots, right? And we use the TI plasmid of that bacterium for gene transfers in many of the cases, as you studied in the second chapter on biotechnology in the applications, that we have used this for transfers related to the interference RNA technology, and in the case of the BT technology also, right? So it's one of the most important, most commonly used gene transfer methods in the plants. So with that, we come to the end of this. I hope this session was a value add for you, and I hope the small things that I pointed out to you to be able to do your paper well is they went down well into your psych. And I will just quickly somehow refurbish that with you for a minute or two. First thing is, play a psychologist. Learn to play a psychologist, because if you learn to play the student, you will not be able to crack it that well, So one tip is that when you are reading a part of the chapter, think like a person who would frame a question from that particular part to confuse. If I tell you, frame a question to confuse even your teachers from this, how would your psychology work at that time, right? So just think of how that psychology works, right? And before attacking the question, before plunging headlong into the question, first please analyze the type of question, right? Read the question and think, what kind of a question does it look like? Does it look like a tag question, which means it is simple with some twist, or is it really simple and direct like the fifth question, right? Or does it look simple but has a very bad, some indirect meaning towards it? So the different kinds of questions must be recognized by you very well, and before you actually hit a tick or a cross or whatever, you need to know what kind of a question this is. So with that, I will come back to you tomorrow with the session on the second chapter in biotechnology, and I wish you all the best.